Heredity. All living organisms reproduce to ensure the continuation of their species. In reproduction, the babies or offsprings look similar to their parents. Do you know why? It is because of inheritance. What is inheritance? Inheritance or heredity is the process by which traits are passed from parents to offsprings. This transfer of traits happens through genes, which are the units of heredity found in the DNA of all living organisms. Genes carry the instructions for the development of specific characteristics such as eye color, hair type, height, and many other physical and biological traits. For example, in humans, a child may inherit the same eye color as one of their parents because the gene responsible for eye color is passed from the parent to the child during reproduction. Similarly, a plant may inherit a specific flower color or leaf shape from its parent plant. However, even though offsprings inherit traits, they are not exact copies of their parents. This is because, along with the inheritance of traits, certain differences also arise. These differences are called variations. Let us see the definition of variations. Variation refers to the differences in the characteristics or traits of individuals within the same species. What causes the variations in organisms? If any changes happen to the DNA, it leads to variations. The changes in the DNA can happen either by mutations or genetic recombination or by environmental influences. Mutation means a sudden change in the DNA. Gene combination means combination of genes from two different organisms. Environmental influences means the influence of certain non-living factors. Among these three, most of the variations are occurred during genetic recombination. Variations can be observed in both the types of reproduction, asexual reproduction as well as in sexual reproduction. Variations in asexual reproduction. In asexual reproduction, the offsprings are produced by a single parent. There is no mixing of genetic material from two different parents. So, the offsprings are genetically identical to the parent. However, even in asexual reproduction, small amount of variations can occur due to mutations, means random changes in the DNA. For example, in a group of sugarcane plants that reproduce asexually, all plants are essentially clones of the parent plant. There will be very little variation among them because they inherit the same genetic material. If a mutation occurs in one plant, it might result in a small change such as a slightly different leaf shape or growth pattern. But overall, asexual reproduction leads to very less variations. Now let us see variations in sexual reproduction. In sexual reproduction, the offsprings are produced by the fusion of male and female gametes, that is sperm and egg cells. Since the offsprings receive genetic material from two parents, there is a greater chance for variations. Each offspring inherits a combination of genes from both the parents, resulting in a unique traits and characteristics. Example: in a group of dogs that reproduce sexually, you will observe a wide range of variations. Some dogs may have different coat colors, sizes, ear shapes, or behaviors. These variations arise because each dog inherits a different combination of genes from its parents. No two offsprings are exactly identical, even though they belong to the same species. Why variations are more in sexual reproduction? The key reason why variations are more common in sexual reproduction is due to genetic recombination. When the genetic material from two parents combines, it creates new combinations of traits. Each offspring receives half of its DNA from the mother and half from the father. This mixing of genetic material leads to the expression of new traits and variations. On the other hand, in asexual reproduction, since the offsprings are clones of the parent, there is very little opportunity for new traits to emerge. Only mutations can introduce variations, and these are very rare. Accumulation of variations during reproduction. Accumulation of variations refers to the process by which small changes or differences accumulate in organisms over generations. When organisms reproduce, they pass on their genetic material to the next generation along with any variations they possess. At the same time, new variations may arise in each generation. Over many generations, the accumulation of variations can lead to significant changes within a population. Importance of variations. Variations play a crucial role in the survival and evolution of species. Some variations are neutral, means they do not have any impact on an organism's survival. However, other variations can be either advantageous or disadvantageous. Variations as a survival advantage. In certain environments, variations can give organisms an advantage, allowing them to survive better than others in their population. These organisms are more likely to reproduce and pass on their beneficial traits to future generations. This process is known as natural selection. For example, imagine a population of bacteria. Most bacteria are sensitive to high temperatures. But due to a genetic variation, one bacterium can withstand higher temperatures. If there is a sudden heat wave, and most of the bacteria will die. But the variant that can survive the high temperatures will continue to live and reproduce. Over time, this variant will dominate the population and its ability to withstand heat will be passed on to future generations. Rules of heredity. The traits are inherited according to the rules of heredity. By studying the rules of heredity, we can understand the patterns of inheritance. Types of traits. The traits are of two types. They are inherited traits and acquired traits. Inherited traits. Inherited traits are the characteristics or traits passed from parents to offsprings. Inherited traits come from the genetic information passed down from parents during the process of reproduction. Inherited traits are not influenced by the environment. They are fixed at birth. These traits bring changes in the DNA of an organism. These traits helps in the process of evolution. Examples of inherited traits are eye color, skin color, hair color, shape of the earlobe, etc. Acquired traits. Acquired traits are the traits that an individual develops during their lifetime as a result of their experiences, behavior, or environmental factors. They are not inherited genetically. The DNA of an organism is unchanged due to these traits. These traits do not help in the process of evolution. Examples: skills like speaking different languages, dancing, playing musical instruments, etc. Rules for the inheritance of traits. Now let us understand how do the traits pass from generation to generation. The inheritance of traits takes place through the transfer of genetic material from parents to offsprings. The father and the mother contribute equal amounts of genetic material or DNA to the child during the process of reproduction. The DNA has smaller units or segments called genes which carry the information about different traits. What is a gene? It is a segment of DNA that determines a particular character or trait. Father DNA has genes for different traits like eye color, type of earlobe, etc. In the same way, mother DNA also has genes for traits like eye color, type of earlobe, etc. That means the offsprings or babies get two genes for each trait, that is one from father and one from mother. For example, the father eyes are brown and the mother eyes are blue. The baby will get both brown and blue color genes. That means for one trait, that is the eye color, the baby will have two genes. This kind of alternate forms of genes for a single character are called alleles. But here, what would be the color of the eyes of the baby? Brown or blue? The eye color of the baby will be brown. Because the brown eye color gene is dominant than the blue eye color gene, so the brown color is expressed out. Dominant trait. The trait that is expressed out when one of the allele is dominant or both the alleles are dominant. Recessive trait. The trait that is expressed out when both the alleles are recessive. This is the basic idea of inheritance of traits. The study of rules of inheritance was begun in 19th century by a famous scientist called Gregor John Mendel. Mendel was the first to make a systemic study of patterns of inheritance of traits in pea plants. He proposed 3 laws of inheritance which are called as Mendel's laws of inheritance. Why did Mendel selected pea plants for his experiment? Mendel chose pea plants which are called Pisum sativum for his experiments for several reasons. 1. Short life cycle. Pea plants have a relatively short life cycle and produce a large number of seeds. This allowed Mendel to observe multiple generations within a relatively short period, enabling him to study inheritance patterns across several generations. 2. Pea plants are bisexual. That means every flower has both male and female reproductive parts. So, self-pollination takes place easily in them. At the same time, cross-pollination also can be done easily by hand. Number 3. Pea plants exhibit clear and distinct traits such as seed color, flower color, and plant height. These traits have 2 distinct forms, that is yellow or green seeds, purple or white flowers, so making it easy to differentiate and observe the variations. Mendel used different sets of contrasting characters of pea plants like plant height, tall or short, seed color, yellow or green, seed shape, round or wrinkled, flower color, that is white or purple, etc. Mendel's experiments. Monohybrid cross. A monohybrid cross is a genetic cross between 2 individuals that differ in a single trait. For example, a tall pea plant and a short pea plant. Mendel selected a pure breed of tall pea plant and crossed it with a pure breed of short pea plant. Pure breed tall means both the alleles for the trait plant height are tall and tall. They are represented by capital T and capital T. Pure breed short means both the alleles for the trait plant height are short. Here they are represented by small t, small t. When these 2 plants are crossed, in the F1 generation, all tall plants are produced. Here we can see all these plants got one allele tall and the second one short. Since tall allele is the dominant one, the plants expressed the trait tallness. So in F1 generation, all plants are tall. Now this F1 generation plants are self-pollinated. Let us understand the outcomes with the help of a Punnett square. The possible combinations of F2 generation are like this: capital T, capital T, capital T, small t, capital T, small t, small t. So here, out of these 4 possible combinations, 3 of them are tall and 1 is short. So the phenotypic ratio of F2 generation is 3:1. Phenotypic ratio means the ratio based on the physical appearance. This is the outer appearance. If we look at the genotypic ratio of the F2 generation, genotypic ratio means the combination of alleles that they have for a trait. Here 1 capital T means both the alleles are of same type, that is tall. So it is a homozygous tall. Homozygous means having both same type of alleles. So it is a pure breed tall pea plant. Next we got 2 capital T and small t's. These are heterozygous tall, means they have one dominant and one recessive allele, one capital T, one small t. Next we have 1 small t. This is homozygous short. Here both the alleles are short. So this is a pure breed of short pea plant. So the genotypic ratio of F2 generation is 1:2, 2:1. Now let us see the dihybrid cross in pea plants. In dihybrid cross, we consider 2 pairs of contrasting characters. Mendel crossed a pure breed of round yellow seeds with a pure breed of green wrinkled seeds. In F1 generation, Mendel got all round yellow seed plants. All of them are heterozygous round and yellow. Now let us observe the F2 generation when the F1 plants are self-pollinated. The possible outcomes are like this. So here in the F1 generation, we got round and yellow 9, round and green 3, wrinkled and yellow 3, wrinkled and green 1. So the phenotypic ratio is 9:3:3:1. Mendel's laws. Mendel's law of dominance. Here is a heterozygous tall pea plant. Means it is a tall pea plant, but it has two different alleles for the character plant height. One tall allele and two short allele. Even though the plant has short allele or gene in it, its tallness is expressed out because the tall allele is dominant than the short allele. This is the first law of Mendel. Mendel's law of dominance states that in a heterozygous organism, one trait will conceal the presence of another trait for the same characteristic. Next, Mendel's law of segregation. The parent cell will have two genes for controlling a character, but when it forms gametes, each gamete will have only one allele or gene for that character. For example, a man has two alleles for his hair color like brown and black in his cell, but his gametes or sperm cells will have either brown or black allele only. This is called as Mendel's law of segregation. During the formation of gamete, each gene separates from each other so that each gamete carries only one allele for each gene. Next, we see the law of independent assortment. For the character color of the seed, the parent has two alleles, dominant allele yellow and recessive allele green. For the character shape of the seed, the parent has two alleles, dominant allele round and recessive allele wrinkled. If the dominant allele yellow is passed to the gamete for color, for shape dominant round or recessive wrinkle, any one allele can be passed to the gamete. That means one allele of a particular character do not influence the inheritance of alleles of other character. This is law of independent assortment. The alleles of two different genes get sorted into gametes independently of one another. How do these traits get expressed? Cells have DNA in the nucleus. This DNA is the information source for making proteins inside these cells. The part of the DNA that has information for making one particular protein is called gene of that protein. That means gene is a segment of the DNA that has information to make a particular protein. For example, we know that plant growth is affected by plant growth hormone. But for the proper production of this hormone, an enzyme is required. This enzyme is a protein and its production is controlled by a gene in the DNA. If the gene is proper, then sufficient growth hormone is produced and the plant will be tall. If the gene is altered and the production of enzyme is less, automatically less growth hormone is produced, which makes the plant short. This is how genes control the physical characteristics. So we understood that each trait is controlled by two genes, but these two genes do not present in the same DNA strand. Our DNA is coiled into the form of chromosomes. Chromosomes are present in pairs, so the two alleles of a specific trait are not present on the same chromosomes. Normally, our cells have two sets of chromosomes, but when gametes are formed, only one set of chromosomes are transferred to them, because gametes are haploid in nature. So out of the two alternative pairs of genes for specific character, only one enters the gamete. When this gamete fuses with the other gamete, then they get the pair of genes for that character. Sex determination. Do you know how the sex of a newborn baby is determined? Sex of a newborn is determined by different factors in different species of animals. In reptiles like snakes and crocodiles, the sex of the newborn will be based on environmental temperature. In case of snails, they can change their sex. So sex is not determined by genes in these animals. But in humans, sex is genetically determined. That means the genes inherited from the parents decides the sex of the newborn baby. Each human cell contains 23 pairs of chromosomes. Out of these 23 pairs, 22 pairs are autosomes and one pair is sex chromosomes. Females have a pair of same kind of sex chromosomes XX, whereas males have a different pair of sex chromosomes X and Y. So females are XX and males are XY. Gametes or sex cells are haploid in nature. That means they have only 23 chromosomes. out of 22 are autosomes and 1 sex chromosome. In males, when the gametes are formed, only one sex chromosome is passed to them. It can be either X or Y. So some sperm cells are formed with X and some with Y. Whereas in case of female gametes, all are X. So all egg cells carry X sex chromosome. During reproduction, if the sperm cell with Y sex chromosome fuses with the egg cell, it forms a zygote with XY sex chromosomes. XY means the baby will be a boy. If the sperm cell with X sex chromosome fuses with the egg cell, it forms a zygote with XX sex chromosomes, means the baby will be a girl. Thus, the sex of the child will be determined by the chromosomes that they inherit from their father. This is all about the heredity in why do organisms reproduce. Organisms reproduce for two main things. 1. To maintain the survival of their own species. 2. To introduce genetic variations in their population. What are genetic variations and why they are important? The genetic variations are very important for the adaptation to changing environment. For example, there is a bacteria existing in the oceans near the poles, where the water is usually cool. These bacteria produced a variant which can withstand high temperature. Suddenly, the temperature of the ocean increases due to global warming. Now, this variant which can withstand high temperature survives and the other bacteria becomes extinct. In this way, variations help the organisms to survive in a changing environment. Types of reproduction. Reproduction is of two types, asexual reproduction and sexual reproduction. First, let us learn about asexual reproduction. Asexual reproduction means the reproduction that takes place without the fusion of sex cells or gametes. Only one parent is involved in this type of reproduction. So, asexual reproduction is a process that involves a single parent and no fertilization or gamete formation takes place. Asexual reproduction is observed in both unicellular and in multicellular organisms. First, let us see the different modes of asexual reproduction in unicellular organisms. Binary fission. Binary means two. Fission means splitting. In this mode of reproduction, one parent organism splits into two daughter organisms. This type of asexual reproduction is observed in single-celled organisms like amoeba, bacteria, and euglena. Here we have two unicellular organisms. This is amoeba and this is leishmania. Leishmania causes Kala-Azar disease in humans. Amoeba has no fixed shape to its body. It can change its body shape. The fission in amoeba can take place in any plane because it has no specific shape, whereas leishmania splits in a specific direction since it has a specific shape. Multiple fission. Multiple fission means splitting into many. That means one organism splits into many new organisms. Certain single-celled organisms like Plasmodium reproduce by multiple fission. Next one, budding. Yeast is an unicellular fungus. They produce buds on their bodies. These buds get separated and grows into an individual organism. This mode of reproduction is called budding. These are the methods of asexual reproduction in unicellular organisms. Now let us see the methods of asexual reproduction in multicellular organisms. 1. Fragmentation. Spirogyra is an algae. It is a multicellular organism with simple body organization. It reproduces by fragmentation. In this method of reproduction, first the organism breaks into fragments and each fragment grows into a new organism. Next, regeneration. Certain multicellular animals have an ability to reproduce from their cut body parts. For example, planaria and hydra. If these organisms are cut into pieces, each piece can grow into a complete individual. This is called regeneration. Next, budding. Hydra can reproduce by another sexual method of reproduction called budding. Hydras have regenerative cells in their bodies. They use these cells to produce small buds on their bodies. These buds grow in size and becomes big. They get separated from the parent organism and grows into an individual organism. Vegetative propagation. Flower is the reproductive part of the plant, whereas stem, root, branches, leaves and buds are vegetative parts of the plant. If plants reproduce through their vegetative parts, it is called as vegetative propagation. Cutting, layering, and grafting are the three major techniques of vegetative propagation. Sugarcanes are grown by stem cuttings. Roses can be propagated by layering. Many fruit plants and trees are grown by grafting techniques. Advantages of vegetative propagation. 1. Plants grown through vegetative propagation can flower and fruit sooner than those grown from the seeds. 2. These techniques enable the propagation of seedless plants like bananas, oranges, roses, and jasmine. 3. Plants produced through vegetative propagation exhibit genetic similarity to their parent plant, ensuring they inherit all of its characteristics. Next, reproduction through leaves. In bryophyllum, certain buds are produced in the notches of their leaves. These buds are called epiphyllous buds. When these buds touch the soil, they germinate into new plants. Next, spore formation. Rhizopus, a common bread mold, reproduces through a process called sporangiospore formation. Specialized structures known as sporangia develops at the tips of the upright filamentous structures called hyphae. Inside these sporangia, numerous asexual spores called sporangiospores are produced. As the sporangium matures, it eventually ruptures, releasing the spores into the surrounding environment. These spores can then disperse and germinate under suitable conditions, giving rise to new rhizopus fungi. This is all about the asexual reproduction in organisms. Sexual reproduction in plants. Flower is the reproductive part of the plant. These are sepals. These are petals. This is pistil. Pistil is the female reproductive part of the flower. And this is stamen. It is the male reproductive part of the flower. A flower that has both male and female reproductive parts is called bisexual flower. Example, hibiscus, mustard. A flower that has only either male or female reproductive parts is called as unisexual flower. Examples, papaya and watermelon. Now let us study the reproductive parts of the flower. Stamen has two main parts named as anther and filament. Anther consists of small reproductive units called as pollen grains. If we touch the stamen of a flower, some yellow-colored dust-like material sticks to our fingers. It is nothing but pollen. These pollen grains carry male reproductive cells in them. Pistil. It has three main parts: stigma, style, and ovary. The top part of the pistil is called stigma. It is sticky in nature. The middle long tube-like part is the style. The bulged bottom part of the pistil is ovary. Ovules are present inside the ovary. Each ovule has an egg cell in it. Pollination. For the process of fertilization, the pollen grains has to reach from anther to stigma. This transfer of pollen grains from stamens to stigma is called pollination. If the transfer of pollen grains takes place between the stamen of a flower to the stigma of this same flower or another flower on the same plant, it is called self-pollination. If the transfer of pollen takes place from the stamen of one flower to stigma of another flower on different plant, is called cross-pollination. The pollen grains are carried to the pistil by the help of different agents like wind, water, insects, or animals. Fertilization. Once the pollen lands on the sticky stigma, the pollen starts germination and produces a long tube called as pollen tube. This tube travels through the style and reaches the ovule present inside the ovary. The male cells from the pollen grain enters the ovule through this tube. The male cell fuses with the egg cell present inside the ovule and forms a zygote. This process is called fertilization. After fertilization, the zygote divides several times and forms an embryo. The ovule develops a tough covering around it and it turns into seed. the ovary grows in size and gets ripened to become a fruit. That means after fertilization, the ovule of the flower becomes seed and the ovary becomes fruit. Remaining parts like stigma, style, petals, sepals get dried up and fall off. The seed contains the embryo, and when it gets the necessary conditions, then it germinates and grows into a new plant. This is about the sexual reproduction in plants. Now, let us learn about sexual reproduction in human beings. Puberty. Human reproduction is sexual type. For sexual reproduction, sex cells or gametes are required. These gametes are produced by the reproductive organs. Male reproductive organs produce male gametes and female reproductive organs produce female gametes. Boys and girls possess reproductive organs by birth, but they do not produce gametes till they attain certain age. The maturation of special tissues present in the reproductive organs takes place between 8 to 13 years of age in girls and 9 to 14 years of age in boys. At this age, along with the production of gametes, certain bodily changes also begin to happen in boys and girls. This is called beginning of puberty. During this process of puberty, a child grows into a complete adult. Puberty ends by 18 to 19 years of age. Puberty is the process of physical changes through which a child's body matures into an adult's body and becomes capable of sexual reproduction. Now let us see what kind of changes takes place in the bodies of boys and girls during puberty. Changes those are common in both boys and girls. 1. Growth of thick hair on new areas of body parts like in armpits and in genital areas between thighs. 2. The skin in these areas becomes darker in color. 3. Growth of thin hair on hands and legs as well as on the face. 4. The skin becomes oilier and may be prone to acne due to increased sebum production. Acne is the common skin condition during puberty. Changes that takes place only in boys during puberty. 1. The voice becomes hoarse and cracked. The Adam's apple becomes more prominent. 2. Thick hair grows on the face and forms mustache and beard. 3. The penis occasionally becomes enlarged and erect. Changes that takes place only in girls during puberty. 1. Breast size begins to increase. 2. The area near the nipples of the breast becomes darker in color. 3. The hips become wider. 4. Menstruation or periods begin. How do these bodily changes helps in the process of reproduction? Growth of hair on face and body parts acts as an indication for sexual maturity. Means a sexually matured animal identifies another sexual matured animal for mating with the help of these indications. Mating means the joining of male and female bodies for the deposition of male cells into the female's body. The enlargement and erection of penis helps in the deposition of sperm cells in the female reproductive organs while mating. The hips of the females become wider to facilitate pregnancy and delivery of the baby. The breasts grow in size and matures to feed milk to the newborn babies. This is all about puberty. Human male reproductive system. Human male reproductive system perform three important functions. 1. Production of male gametes or sperm cells. 2. Secretion of male sex hormone, that is testosterone. 3. Deposition of gametes in the female reproductive system. There is a pair of testes present in the male reproductive system. The production of male gametes and secretion of male sex hormone is done by testes. Testes are present outside the body cavity. They are located in an elastic sac-like structure called as scrotum. Testis produces the male sex hormone, that is testosterone. This hormone helps in the puberty of males. This hormone is responsible for the bodily changes that takes place during puberty. Why the testes are hanging outside the body cavity? Because testes cannot produce sperms at the body temperature. They need a low temperature to produce sperms. That is why they are present outside the body cavity. The sperm cells produced by the testes are small and motile, means moving cells. Each sperm cell has a headpiece middle piece and a tail. Now the sperms produced by the testis are to be deposited in the female reproductive system to reach the egg cell. Sperms need some accessory materials for the successful journey to the egg cell. Sperms produced in the testis travel through a duct called as vas deferens. At this point, the secretions from seminal vesicles and prostate gland are added to the sperms. These secretions and sperms, together called as semen, these secretions make the movement of sperms smooth and easier. They also provide nourishment to the sperms. This semen passes out through this duct called urethra, which is present inside the penis. Urethra is the common duct for the passage of sperms and urine, but both do not pass at the same time. During mating, the semen, sperms and secretions are deposited in the vagina of the female reproductive system by the penis of the male reproductive system. Human female reproductive system. Female reproductive system performs 4 important functions. 1. Production of female gametes or egg cells. 2. Secretion of female hormones, that is estrogen and progesterone. 3. Providing necessary conditions for the fertilized egg to develop into a baby. 4. Menstruation. First, let us see the production of female gametes or egg cells. This task is done by the ovaries of the female reproductive system. By birth, girls contain a pair of ovaries. Each ovary contains thousands of immature eggs. On reaching puberty, these eggs start maturing and gets released. Each ovary alternately releases one egg approximately every month. The released egg is collected into a tube called as fallopian tubule. The sperms deposited by the male in the vagina during sexual intercourse travels through the cervix, uterus and reaches the egg present in the fallopian tubule. If the sperms fertilize the egg, it becomes zygote. This zygote divides repeatedly and forms an embryo. This embryo moves towards the uterus and gets implanted in the wall of the uterus. The lining of the uterus grows new tissues and blood vessels to accommodate the embryo. After implantation, the embryo grows into a fetus. The embryo gets nutrition from the mother's blood with the help of a special tissue called placenta. This is a disc-like structure embedded in the walls of the uterus. This placenta has blood spaces on the mother's side and villi on the fetus side. This arrangement provides large surface area for the exchange of materials between the blood of the mother and the blood of the baby. Here, the nutrients and oxygen are absorbed and wastes are released into the mother's blood. The development of baby inside the mother's womb takes approximately 9 months. This period is called gestation period. The delivery of the baby takes place by the rhythmic contractions of the uterus. What happens if the egg is not fertilized in the fallopian tubule? Every month, the walls of the uterus get lined by blood vessels and tissues in the anticipation of embryo. If the egg is not fertilized and the embryo is not formed, then the unfertilized egg lives for 1 day. After that, the egg gets dissolved and the tissues formed on the lining of the uterus along with the blood slowly breaks and expels out through the vagina. The loss of blood and mucus from the vagina lasts for 2 to 8 days. This process is called menstruation. Since this process is periodical and happens for every 28 days, it is called as menstrual cycle or periods. This is all about the female reproductive system. Reproductive health. A boy has got the capability of producing sperm cells, and the production of ovum started in a girl. Does it mean that they both are ready for sexual act to produce babies? No. The mind and body should be completely matured to participate in sexual acts. Otherwise, one needs to face so many complications. Health complications due to sexual acts. Sexual act involves the intimate contact of body parts, which may lead to the transmission of certain bacterial diseases like gonorrhea and syphilis and viral infections such as warts and HIV/AIDS. For girls, getting pregnant at younger age can adversely affect their health. How to avoid these complications? 1. Participating in sexual acts only when the body and mind are completely matured, that is, only after crossing 18 years of age. 2. Having sexual relations with a single trustable partner. 3. By using contraceptive devices and methods. Contraceptive devices and methods. Condom. Condom is a rubbery covering that is worn on the penis during intercourse to prevent the entry of sperms into the female reproductive system. It also prevents the transfer of germs between the partners and give protection from sexually transmitted diseases. Copper T. Copper T is the other contraceptive device that is placed in the uterus of the females to prevent the fusion of sperm cell and egg cell. This device may cause irritation in some people. Next, contraceptive pills. These are the drugs that change the hormonal balance in the female body and stop the release of eggs. The prolonged use of these pills can cause side effects. Next, surgical methods. The vas deferens of males is cut and ligated to prevent the release of sperms. In the same way, the fallopian tubules of females are cut and ligated to prevent entry of eggs. These are the permanent methods. But if the surgery is not done properly, it may lead to infections. What if the contraceptives fail and pregnancy happens? Medical termination of pregnancy. Using surgical methods to remove unwanted pregnancy is called medical termination of pregnancy or abortion. In certain sections of our society, sex-selective abortion of female fetus is practiced. This leads to the disturbance of male-female sex ratio in the population. What did the government do to protect the girl child from these false practices? The government has banned the prenatal sex determination to control the misuse of medical termination of pregnancy. Prenatal sex determination means scanning and identification of gender of the fetus. This is all about how organisms reproduce. Thanks for watching. Please like the video. Please share this video with your friends. Please subscribe to Grade Booster channel. Press the bell icon to get all the latest updates. Check the description to find links of other useful videos.